Researchers at Trinity College Dublin, working together with the Royal College of Surgeons in Ireland, RCSI, have developed special fluorescent, color-changing dyes that, for the first time, can be used to simultaneously visualize multiple distinct biological environments using only one singular dye. When these dyes are encapsulated in delivery vessels, like those used in technologies like the COVID-19 vaccines, they switch on and give out light via a process called aggregation-induced emission, AIE. Soon after delivery into the cells, their light switches off before switching on again once the cells shuttle the dyes into cellular lipid droplets. Because the light coming from inside the cells is of a different color and occurs within a different time window to the light coming from the same dye inside the delivery vessels, the researchers can use a technique called fluorescence lifetime imaging, FLIM, to distinguish between the two environments in real time. The work was recently published in the leading international journal, Chem. First author, Dr. Adam Henwood, Senior Research Fellow in the School of Chemistry and based at the Trinity Biomedical Sciences Institute, TBSI, worked on this design with PhD student Connie Sigurvinsson. Dr. Henwood explained, bioimaging relies on on-off dyes where the dyes only emit light under one set of conditions but are otherwise switched off. We do this by timing how long it takes for the light coming from our samples to reach the microscope. Light from the delivery vessels takes marginally more time than light from within the cells. The time differences are small, just a few billionths of a second either way, but our method is sensitive enough to capture it. This unique quality means the dyes could have a huge suite of applications and, for example, hold the potential to revolutionize biosensing and imaging approaches. Because these dyes can help scientists map the intricate structures within living cells with such high contrast and specificity, they could help illuminate how drugs are taken up and metabolized by cells or allow scientists to design and conduct a range of new experiments to better our understanding of the complex inner workings of cells and their all-important biochemical machinery. In the published journal article, the scientists focused on using the dyes to image cellular lipid, fat, droplets, which are one example of important organelles that make up living cells in most complex organisms. Lipid droplets, once considered to be simple fat reservoirs, are now believed to play an important role in regulating cellular metabolism, coordinating lipid uptake, distribution, storage, and use in the cells. One potential avenue of further research is to see whether the team can target other important cellular organelles with their dyes. Thorfinnur Gunnlaugsson said, being able to monitor cellular function or the flow of molecules or drug candidates within cells by observing different fluorescence emission colors is extremely attractive. The breakthrough here is that we can resolve and use the difference in their fluorescence lifetimes to identify these same probes within different cellular environments in a fast and accurate manner, literally allowing us to map out their colorful time travel within the cells. Most exciting, however, is that this phenomenon does not apply to cellular imaging. They envisage many new possibilities for these dyes, pointing toward their exceptional sensitivity as attractive for developing sensors of hazardous environmental pollutants or using their bright, light-emitting properties to power chemical transformations, analogous to nature's own photosynthesis. The research has both an international, 8 nations are represented, and Irish feel to it, with the latter's key funding bodies, the Irish Research Council, IRC, and Science Foundation Ireland, both playing critical financial support roles. The closer we look at the molecule-cell interface, and crucially, the better we can see, in real time, how molecules diffuse from place to place inside the cell nanomachinery, the closer we get to realizing Richard Feynman's dream of understanding everything that living things do from the wiggling and jiggling of atoms. But only recently have researchers had sufficient experimental and computational resources to track these motions and vibrations in complex biological environments. This exciting new work demonstrates more specific, high-contrast imaging of subcellular dynamics, which will in turn enable researchers to develop more effective drug formulations with reduced side effects.